nanoscopy. So the work I will present today has been done in collaboration with Stefan Hell, with Damián Refojo from Buenos Aires, with Alfredo Cáceres, Nicolás Unzain, and Mariano Bisbal from Córdoba in Argentina, and with Guillermo Acuña from the University of Freiburg in Switzerland. Okay, so all this, we're gonna skip it. How good is fluorescence nanoscopy? We all know. We know what's good for, what the limitations are. We know about the diffraction limit. So I will just want to stress the size mismatch that we have between our photon sources, between our fluorophores, our molecules, 1 nanometer in size, roughly, and the image of such a fluorophore in a microscope is about 200 nanometers. So we have a size mismatch of 1 in 200. It's a very large size mismatch, so there's a lot of information that we miss if we do conventional imaging. Okay, so let's move on. So fluorescence nanoscopy was invented. A first generation of methods provides a resolution in a few tens of nanometers. There are 2 families of methods. Luca just mentioned the coordinate targeted nanoscopy and the coordinate stochastic nanoscopy. So how does it work? We are going to skip it. I will do a break. I will stop here just to explain DNA origami, because this is a technology that we use a lot also to test new nanoscopy methods. So how does it work? A DNA origami is basically a way to construct nanoscopic objects by molecular self-assembly using the characteristics, the properties of DNA. So DNA, you know, forms double helix in a specific way depending on the sequence. So DNA origami exploits this property in this way. So we start with a single-stranded DNA that is schematically shown here by this circular DNA. It's single-stranded, usually taken from a virus or something like that. So it's something like 6,000, 8,000 bases, single-stranded DNA. We, of course, know all the sequence of it. And now imagine that we can mix it up with a small oligonucleotide, just maybe 40, 50 bases long, that half of it is complementary to this region, like the green with the green, and the other half of the oligonucleotide is complementary to another region of the long DNA strand, like this one. So red is complementary with red, green complementary with red. If we let these 2 molecules hybridize, then they will form this structure. So the large DNA molecule will fold in this way, trying to accommodate as many complementary bases as possible. Now, if we do this not just with 1 oligonucleotide, but with many, something like 200, 300 oligonucleotides, all of them very precisely designed to fold the original DNA, the scaffold DNA in a particular geometry, then we can do that. And then automatically, by self-assembly, we can form nanostructures with a well-defined geometry. And in addition, we know exactly on this structure the sequence of DNA on each position. And this is how it works. This is, again, a rectangle. These are AFM images of these zillions of rectangles that are formed by a self-assembly. But in addition, as I told you, we know the sequence of each position of this DNA origami. So we can let these short oligonucleotides that we use to fold the large DNA molecule, we call them staples, right? So we can let some of the staples longer. For example, this one, we can leave it here. Can you see my cursor? No, I can make a laser pointer. Okay, so for example, this staple, we can leave it at this position a little bit longer with an extra DNA sequence that we can use afterwards to bind at that particular position a fluorescent molecule, for example, or a nanoparticle. So the DNA origami, it can not only form these funny nanostructures, but it can be used, we use them as nano breadboards, like nano optical breadboards to put here light sources, nano antennas, or different components to produce different optical effects. For nanoscopy, we will place basically fluorescent molecules in organized arrangement. Okay, let's move on. So here you see an example of that. So we have, this is a DNA origami that is has a cylindrical shape, and we can leave these black DNA chains are like single-stranded DNA that we leave protruding out of the nanostructure on specific regions. And we do it like to provide spots for DNA paint. So these are spots where other fluorophores or other oligonucleotides, now fluorescently labeled, can transiently bind to these short DNA chains. And in this way, they can, they come here, they bind for a while, we can make an image, they go, then they bind somewhere else, they go. And in this way, we obtain the blinking necessary to do a super resolution image. And for example, this structure can be used as a calibration sample for super resolution microscopy. Okay, this is an example of this structure. You get the blinking that you need to do stochastic nanoscopy. And if you analyze this, then you get this video is converted into this image where you get all these, each one of these bright spots is in fact 3 spots organized in a line and with the separation distance according to the DNA origami design. Okay, so targeted nanoscopy has been already explained. So let's move on. So here, let's make just one conceptual note. So in this kind of nanoscopy, like wide field, right? We detect each emission spot and we extract all the information from the emission. So all the illumination is uniform. So we are not injecting any information into the problem with the illumination. We illuminate with a uniform intensity. There's no information there. We collect all the information from the emission of the molecules. We get these emission patterns, we can fit them, for example, with a Gaussian function, and can determine the center. To do this properly, we need a lot of photons to have a good fit and to have a good localization. That's the limitation of stochastic nanoscopy. We need a few thousand photons to get into a 10 nanometer resolution regime. In coordinate targeted nanoscopy like STED, the situation is kind of the opposite. Why? Because we are injecting all the information with the illumination. No? We have this kind of illumination pattern where we have a excitation focus and the depletion focus, and we are going to move these 2 laser foci over the sample with a piezoelectric scanner, for example. And then we can stand in one position, wait a certain time, and ideally, if I collected 1 photon, I could say with some statistical certainty that at that position there is a fluorescent molecule. And then I can move this pattern to another position, wait a certain time, and if I collect a photon, I can say, okay, there is a molecule here, or not, with some statistical confidence. That would be the case if there would be no background, for example. Because each photon I detected, I can assign it to a fluorescent molecule. But in real life, there's always background. And in real life, also this depletion laser is never perfectly efficient. So at the end of the day, again, we need a strong fluorescent signal to claim that there is a molecule at that position, because it has to emit above the background. And so both techniques end up being limited by the photostability of the fluorophores. That's, I mean, these techniques, they don't have a theoretical limit. They have a practical limit due to the limited budget of photons that we have from each single molecule. So for that reason, it makes sense to try to come up with some idea to detect molecules in a more efficient way. So this first generation of methods can deliver a lateral resolution that is between 15 and 60 nanometers, and an axial resolution that is about 3 to 4 times worse, in the 30 to 120 nanometer resolution. So nevertheless, this is wonderful. You know, you can see a lot more than before. Like here, you see these microtubule bunches, and that when in a diffraction limited image, you cannot discern very well where is one microtubule from the other. when you do nanoscopy, you can now see very nicely the pathway of each microtubule. But again, as in confocal microscopy or normal microscopy, if we zoom in one of these microtubules, we get this kind of images. They get blurred again because we are at the limit of the resolution of these methods. And so we would like to see better than this, because we know that microtubules are actually a supramolecular protein structure, a quasi-crystalline structure of tubulin, of alpha and beta tubulin, forming this quasi-helical structure. So there is organization there, and we cannot see it. We cannot see it. We again don't have the necessary resolution to see the molecular organization. And we would like to do that. And to do that, we need another push into the resolution. We need to get really into the sub 10 nanometer regime, and then we will get really in a resolution regime that is comparable to the size of the structural proteins, and probably we can start seeing these kind of structures in the cells. So that's what we want to do. Okay, so this I told you, let's move on. So one way to do this is a method called Influx. Influx is you will see it's actually a very simple method conceptually. Instrumentally, it can become complicated, but conceptually, it's a kind of triangulation problem. So what we are doing is illuminating, we are combining the injection of information with the illumination with single molecule localization. So we are illuminating the sample with some excitation field that is position dependent. Let's see it first in one dimension. We want to locate this molecule, this green star. We want to locate it on this dimension X. So we illuminate this with a field that depends on position. And then when the molecule is here and we use this excitation field, we will record a certain number of photons, N1. Now we switch off this field and we switch it on, displaced along the direction we want to locate the molecule. Now the molecule is excited with a different intensity, in this case higher, so we will record a larger number of photons, N2. So you will see that now N1 and N2 are encoding the position of the molecule. So we can actually establish an estimator that will give us, based on N1 and N2, will give us with maximum likelihood, for example, the position X of the molecule. That's the way Influx works. The only particular thing about Influx is that we discovered that using zeros of intensity, using excitation fields with zeros of intensity instead of maxima or other shapes, using zeros of intensity, you become very, very photon efficient. So you can detect the position of the molecule with a minimum number of photons. Okay, so you can establish now the statistics of this is, of course, a binomial probability distribution. And then you see that here in light blue, you see the P, the parameter of the binomial distribution that has this shape. So of course, this method is limited in some sense. It works in a one-to-one fashion only between the position of the two zeros. So if I know the molecule is between here and here, then P goes from 0 to 1. The problem is well defined and I can determine the position. If I cannot assure that and the molecule can be outside this region, then the problem is not well defined because I have two positions that could have the same likelihood estimate. So in some way, Influx is sacrificing a field of view to provide a very high efficiency in the number of photons to locate the molecule. So, okay, you can extend this to multiple dimensions, of course. And in practice, for example, to do it in 2 dimensions, it turns out that you need 4 zeros of intensity to do this the most efficiently. And how do you implement this on a microscope? Well, the first way this was done was like this. So in principle, this looks like a confocal microscope, but with some modifications. First, you need this VP. This is a vortex plate to introduce a phase modulation in the beam in order to produce this focus with a 0 of intensity at the center, like instead, the same way. But now we are using it for excitation, not for depletion. We are exciting the sample with this donut-shaped focus. And the other thing we have to do is to shift this focus between these 4 positions. And in this case, this was done with an electro-optic scanner. So basically, first you illuminate the sample with a 0 at this position, you record some number of photons, you switch it off, you switch it on a different position, here for example, you collect a second number of photons, you switch it off, switch it on here, again you record a different number of photons, you switch it off, you switch it on here, and you get the 4th number of photons for this molecule. And now with these 4 numbers, N1 to N4, you can estimate the position of this molecule. That's it. The complications of this setup is that you have to do this very fast, right? Because if you want to make this compatible with the blinking, then you need to shift these positions very fast. That's why we used an electro-optical scanner in this case. So I told you this is very efficient. Here is the proof. These are the simulations, the Kramer-Rao lower bound to locate a molecule inside this area using just 500 photons. And you see that inside the mean flux field of view, so to say, the precision is between 1 and 3 nanometers. Very, very good precision with 500 photons only. And of course, we can do this experiment by moving a molecule with a piezo scanner, for example, and compare the set position with the measured position. And this is the result on the same color scale. So indeed, it is very efficient. You can locate the molecule with just 1 or 2 nanometer precision inside the field of view. The localization error is not isotropic due to the geometry of the field. Okay, but these are technical details I can discuss if somebody's interested. And of course, it's also position dependent. So it's very, very good near the center, and it becomes worse as you reach the borders of the excitation field. But in all cases, it's very, very precise. Here you have, for comparison, the Kramer-Rao bound for a camera-based single molecule localization with a signal to background ratio of 50, which is actually very, very hard to reach in a real experiment. And this is like 2 times better with the same number of photons. Another way to show this is in this kind of plots, like the localization precision as a function of the total number of photons. And we still have the typical inverse law with the square root of the number of photons, like in a camera base, but mean flux provides an additional knob to the experiment. So we can, for example, if we use a field of view of 150 nanometers, we get this curve, way better, like we can get like a 3 nanometer precision with 1,000 photons. But if that's not enough, we can reduce the field of view. And with the same number of photons, we are much better. So mean flux is a tool that is adaptive and is highly precise to look in regions like smaller than the focus. So if we really want to look inside the focus, you can use a mean flux to do that. And it's a technique to locate molecules. So you can use it either for nanoscopy, if you combine it with blinking. For example, inside here, if you manage to make the molecules blink, then you can locate each one of them one at a time and then construct a nanoscopic image. Or you can use it for tracking inside this area to make a nanoscopic tracking, or you can do it for tracking over larger scales if you periodically recenter the excitation pattern. So for tracking, it also has advantages because since we need fewer photons to locate the molecule, then we can track faster, obviously. So here you have a performance example using a DNA origami. So for this demonstration, we constructed a DNA origami with this geometry, where we placed 8 molecules separated by distances of 10 to 12 nanometers, a very small origami. And this is the data you collect. So mean flux delivers positions. So you can plot it like this, or you can plot it in this way, like in a false intensity map to make it look more familiar to a fluorescence image. And this is an even smaller origami with distances between 3 and 6 nanometers. And you can clearly resolve those molecules. Okay, And this is an example for tracking inside a bacteria. In this case, we were tracking this protein, the 30S ribosomal protein, fused to a photoactivatable protein, the MEOS2. And in this experiment, we chose to use just 9 photons to locate the molecule. So every 9 photons, we get an estimation of the protein position with a precision of 48 nanometers. So this is very decent precision. So it's not 1 nanometer, but we are using only 9 photons. And that allows us to go very fast. So we get a position every 125 microseconds using fluorescence, using the count rate of a fluorophore, of a fluorescent protein, actually. And that allows us to do this kind of experiments like you see here. We get a diffusion coefficient with a sliding average window of 35 milliseconds. So this is like very comparable to the frame rate of a video camera. But instead of having 1 position, we get like 80 positions. And that allows us to get a diffusion coefficient at video rate. And then we can say, okay, we can see how the molecule is diffusing with a certain speed. Then it gets stuck in 1 position. It remains there for some time. And then it starts diffusing again much faster. And then it gets stuck in a different position and so on. Okay, so that's Influx. That was the first implementation of Influx. It has since then been extended to 3 dimensions with the same concept by the group of Stefan Hell. And recently we just finished building a new implementation of Influx that is experimentally simpler, and it also provides access to lifetime information. And we called it the Pulsed Interleaved Influx, or P-Influx. And it's very simple. You will understand it very quickly. So basically we are doing the same, but instead of scanning this donut-shaped foci with a scanner, with a fast electro-optical scanner, what we do is the following. We just take a pulse laser, we split it in 4, we introduce 4 different time delays between the 4 pulses, and then we recombine them and send them all through the phase modulation vortex faceplate, but we focused each one of these beams on different positions of the sample. And now the excitation pattern of Influx is completed in 1 repetition rate of the laser. So this goes at the maximum possible speed. And we detect each photon with time-correlated single photon counting. So now we know from each detection photon, we can assign each detected photon to which excitation donut, with which excitation donut it was produced. And then we get the information for localization. And at the same time, we get the information for lifetime. To do this, we also developed an open source Python code to control the Influx setup and to analyze Influx data, both conventional and time-correlated single photon counting data from Influx, and also to simulate a full Influx experiment. So, and this is will also be available with the publication. It's actually already available. We are just finishing commenting and making it more friendly to other users. Well, this is demonstration of performance. Of course, it works just as well as Influx. So no, there are no surprises there. You see, this is here we are locating a gold nanoparticle with 50 photons. We get an accuracy of just 10 nanometers. And if we use a few thousand photons, then we get into the 1 nanometer regime as expected. This is single fluorescent molecules. Again, this is a confocal image. If we zoom in there, it looks like that. Now, if we perform Influx there, then it looks like that. I don't know if you can see it, but over there you have a few localizations of Influx with just 1 nanometer resolution. Okay. This is another demonstration of P-Influx to make imaging. So again, in this case, we use again a DNA origami with 3 molecules, and we can locate them with this precision. You see the scale bar here is 5 nanometers. And this technique gives you automatically film, right? It gives you lifetime images. So This is a technique that can really give you lifetime images with molecular scale resolution and with no distortions, because this technique does not distort the lifetime of the molecules like instead. So you really can access the clean lifetime information. And you can use it. This is another example where we tested the pulse mean flux for tracking and multiplexing based on lifetime. So these are two DNA origami structures that are designed to bind transiently to two positions. So this is a long DNA strand with a fluorophore that can bind to this position and to this position transiently. This one, for example, has 8 complementary nucleotides, and that gives us a time of 2 seconds, more or less, 2-second residence time on each position. And this is a faster structure with only 7 complementary nucleotides. So every time it binds, it remains there for half a second, more or less. And okay, we can track these positions in time, like you see here. No, we detect, of course, the 2 positions, but also we use 2 different fluorophores in this case, ATO 542 and Psi3b. So we can discern these 2 structures, for example, based on the fluorescence lifetime or on the average binding time, like you see in these 2 very clear, distinct populations. This was just an example to show the possibility of multiplexing based on lifetime for mean flux. Okay, now I'm going to show you very quickly 2 other techniques that will give you information in the sub-10 nanometer region. This one is called simpler for supercritical illumination microscopy photometric set localization with enhanced resolution. Very complicated name, but what it allows you to do is for stochastic nanoscopy like storm, palm, or DNA paint, you get this kind of images, right, of microtubules. Well, with this simple analysis tool and simple measurement tool, you can get cross sections like this. We can now do cross sections of these microtubules, very clean cross sections, because it provides set resolution that is comparable to the lateral resolution. For storm, we get something like 15, 20 nanometer resolution, and for DNA paint, below 10 nanometer resolution in set. How does it work? It basically consists of using a TIRF microscope in the best possible way, in a quantitative way. So probably you all know TIRF microscopy. What you do is you illuminate your sample with light coming at an angle larger than the total internal reflection, and therefore the excitation light does not penetrate into the sample. You have an evanescent field that decays exponentially like this. Fine, this is well known, and it actually has been used to obtain actual information based on this phenomenon. What we did was to combine this with an additional effect that is also set position dependent, and that is the emission of the molecules. So the emission of molecules close to an interface is also modulated by the presence of the interface. Here, what you see here is the emission angular pattern of a single molecule oriented perpendicular to the water glass interface as a function of the distance to the interface. When it's 250 nanometers away, it looks like that. And as you get closer, more and more light is emitted into the glass space, and therefore into your objective. The same happens for a molecule that is oriented parallel to the molecule, with a different angular pattern, of course, but more and more light is emitted into your objective. This can be calculated. This is the curve for a perpendicular dipole. The blue one is the curve for a parallel dipole, and the red one is the isotropic average for a molecule that is rotating during our integration time. So you see that in the end we have 2 effects that make molecules closer to the interface brighter than molecules farther from the interface. So what we did was basically to compute these 2 effects carefully, and it looks like this. So we can normalize this to the emission of a molecule at the interface. That's the N0 is the number of photons of a molecule placed exactly at the interface. So the number of photons of a molecule at any axial position normalized to a molecule at the interface has this follows this function. In red, you see the exact solution, and in blue, you have an exponential fit to it. So it's almost exponential. And for practical reasons, you can use just the exponential approximation, which has this shape. So basically, if you know these parameters, df and alpha f, and you measure the emission of a molecule at the interface, this can be measured, N0, simply by throwing, for example, labeled antibodies on the cover slip and measure their intensity, like here. This is the histogram of emission intensity of molecules just spread on a glass cover slip. So just with these three parameters, you can compute from one measurement of intensity, you can compute the axial position of molecules. And this is what is done, because in stochastic nanoscopy, what you do is you record images of molecules with a camera, and then you have single molecule emission events like this one. A molecule switches on, emits for some frames, and then it switches off. Well, the only care that we are taking is that we are discarding the first and the last frames of this event, because during those first and last frames, we cannot be sure that the molecule emitted during the complete integration time. So we discard these two, and from the rest of the frames, we compute an average number of photons. And with that number of photons and the calibration that I just showed you, we directly assign an axial coordinate to that molecule. The lateral coordinate is obtained, of course, from the Gaussian fit. So with the same kind of data and with a simple calibration, you can get a 3D image of this quality that is actually only rivaled by 3D Influx or very sophisticated 4-pi setups. Okay, so if we compute the resolution, we have a better resolution that we have sub 10 nanometer resolution in the 3 dimensions. And I showed you that one would like to, for example, see the structure of microtubules, right? Now we have the resolution to do it. And this is a cross-section of a microtubule. If we try to identify this kind of helical structure, it's not possible. It's not there. But it's not a problem of resolution. It's a problem that the labeling is not dense enough. So placing, if I really want to resolve this, I need to place one fluorescent molecule every 8 nanometers. And that's not simple. I mean, due to electrostatic or steric hindrance, this is usually impossible. But nevertheless, we can compute statistics, for example, of distances to the first 5 neighbors in the XZ plane or in the XY plane. And if we do that, we see histograms peaking with a separation of about 8 nanometers in both cases, showing that even though the labeling is not perfect, the resolution is enough, and we can even indirectly tell that there is underneath there is a structure as expected. Or in cases that we don't know, we can even start inferring whether there is a structure or not at the molecular scale. Okay, what's the time? I think I still have a few minutes. So I'm going.
You still have, I will say, around 5 minutes.
Okay, perfect. So these slides, I just pasted them during Luca's talk. So they are not very well organized, but I wanted to share with you our latest experiments on STED-FRET. So you all know STED by now, and you all know, of course, FRET. And it's interesting, it would be nice to combine them because FRET provides, again, sub 10 nanometer information, and STED provides information in the, let's say, 20 to 60 nanometer range. So when one does FRET imaging, one is obtaining information about molecular interactions, but it's impossible to say where those interactions are, because the information is an average over a diffraction limited spot of 200, 300 nanometers. And so we obtain average information, which is in many cases is useful and very nice, because it's good to have an average information. But if one wants to know where these molecular interactions are placed and what's the heterogeneity of it, that's not possible. And that's the reason why this is something that has been identified from the beginning of the super resolution microscopy. I mean, the advantages of combining FRET and super resolution, there are papers written about it, but so far there is no to our knowledge, no method of, let's say, general applicability to combine these two techniques. And we believe that this can solve this problem. So basically what we did was to combine STED and FRET using an intensity-based measurement. And I will show you. The setup is basically a two-color STED setup. There's nothing new here. So it's compatible with any two-color STED. The first thing we did was, okay, we used DNA origami again to test that this was working. We used an origami like this one. It's a cylindrical structure with two spots separated by 180 nanometers, two FRET spots. Each FRET spot has two donors and one acceptor. You may wonder why two donors and one acceptor and not just a single pair. Well, the reason was simple, because this donor was bleaching too fast. So we wanted to be as close as possible to a single pair FRET, but also be able to collect statistics in a reasonable rate. So we just put two donors. In this case, the donor was ATO 594 and the acceptor was ATO 647N. Two dyes that are suitable for STED. And also they have a reasonable overlap to do FRET. So what you see here is the two-color imaging, confocal and STED. Maybe in the STED you cannot see very clearly. Every single spot in the confocal image is a two-spot image in the STED image. You can see here below some single origami structures imaged by STED in the two channels. And this is the average of these four, for example. So clearly we can resolve the two FRET spots. And then we can take a look if there is FRET going on. So we can do confocal FRET, of course. And you see, we clearly see anti-correlated signal jumps between donor and acceptor. These are kind of complicated anti-correlated jumps, because remember we have two donors and one acceptor, but still you can see FRET going on. But sorry, and here you have two pairs of FRET at the same time, because they are inside the diffraction limited spot. And here you see STED FRET super resolved. Here we have the images, and now we basically what we do is we take several consecutive STED images, in this case 16 STED images, and then we can follow the intensity, for example, of this one, of this spot, and you see clearly anti-correlated jumps. Or in this case we follow this one, and again you see anti-correlated jumps and so on. So this proved us that we can super resolve FRET events. Now we needed a method to apply this in real samples. And okay, these are simulations. Maybe I don't have time to show you the details, but the formalism of FRET that we use to compute FRET is this one developed by Tom Jovin based on intensity and the so-called KT over KF method. It's basically a computation of intensities. You need to record three images: the image of acceptor emission and the donor excitation, the image of donor emission excitation, and the image of acceptor and the acceptor excitation. And all the other parameters are just corrections due to the setup that measure the crosstalk and so on, and the efficiency of excitation of each molecule. So here you see an example on microtubules. What we did here was just to label microtubules with the donor and acceptors all over it, just to have a subdiffraction scaffold sustaining FRET, and see if we can resolve that. And that's what you see. Here you see the microtubules super resolved by FRET, and you see by STED, sorry, and you can detect FRET on them. The other thing I want to show you here are these histograms. So it is important for this method that even if there is no FRET, just by fluctuations of intensity, you will compute some F value or some FRET efficiency value. And this becomes more critical when you do it on subdiffraction regions, because you have fewer molecules. So you have just a few donors, just a few acceptors. So if you have one or two missing, the ratio of intensities will change, and then you will have some distribution that, let's call it a a background distribution of F values. And this is the histograms that we call negative control here. We computed that on the microtubule sample, for example, doing samples labeled only with donor, samples labeled only with acceptor. Then we align artificially two microtubule segments, and we compute the F due just by random intensity fluctuations. That's that histogram. And then when you have real FRET, you have these other histograms, the ones on they call in blue. So it's important to measure both, and then you can set up a threshold. And of course, you will sacrifice some positive FRET sites, but the ones you detect in the end are clearly positive FRET and with a predefined statistics. Okay, so that's how it works. And finally, here we showed some examples on the MPS, the membrane periodic skeleton of neurons. This is a structure composed of adenosine, actin, and spectrin, and we can show that FRET takes place exactly over those rings, even though actin is distributed all over the cell. The FRET sites are located only, the FRET sites with adenosine are located only on the MPS structure, which is only resolvable with super resolution. Okay, with that I finish, and I'm happy to answer any questions that you may have. Thank you very much.
Thank you, Fernando, very much.